future. So basically today, we're going to talk about flow cytometry, insights, and applications. So we're going to talk is about a few things that are based in the flow cytometry. First of all, I would like to explain what is a cytometer. So we are going to do a very basic concept overview about what is methodology, how it works, basically. Then we're going to talk about the components of a cytometer, because all these elements are necessary to make this instrument works properly. How to define the concept of how it works, basically, in terms of detection, trying to understand much better how this instrument can be applied to different science or medicine, industry, or whatever. The main function of the cytometer, depending of the field. Some applications of flow cytometry, particularly mostly in medicine, which is the most predominant application. Then we're going to talk a little bit about the industry applications, ecology as well, and even in the space, which is also quite trending topic at the moment. And then at the end, we can discuss any potential questions that you have. I would like to do a summary about this presentation. So basically, the cytometry is the etymology of this word comes from the Greek. So that means cyto, this word means cell, basically, and metry means measurement. So basically, the cytometer, in few words, is an instrument which is able to measure cells or properties of the cells. So today, up to date, the methodology of flow cytometry is considered as an instrumental methodology that allows the quantification and characterization of microscopic particles or cells in suspension. So basically, when we get any potential sample from a hospital or any biological sample, all these samples, they are usually composed by many cells. Here you can see in this representation of a tube, we have many cells by different colors. That means that every cell has different properties, and this cytometer is able, through a multidimensional analysis, which means that we can analyze different parameters at the same time, all these cells in an individually manner. So how we do this? Basically, what happens is when the cells are acquired by this kind of instrument, all the cells are aligned into a single column. So when the cells arrive to a very specific position, which is called interrogation point, the cells are exposed to a laser. So when that happens, the cells, all the information that we generate when the laser is exposed, or the cells are exposed to the laser, is collected by an optical system, basically, which is composed by filters, mirrors, and detectors. So in that moment, in this interrogation point, we can get information about the size of the cells, the complexity, and also if these cells has some kind of fluorescent properties. This laser normally is located in an orthogonal position. That means it's 90 degrees from the acquisition, and the cells are aligned thanks to a stream that we put inside the machine. So we make all the heterogeneous cells goes and run like a single column. Then when the cells are already past the interrogation point, they are still, in the single column, but they are going to the waist of the instrument. So every cytometer is composed by 3 different systems. These 3 key systems are essential, and if one of them is not working, the machine is not able to analyze the cells. The first one is the fluidic system. This system is composed by different kinds of pumps and pressure controls that allow the fluids going into a laminar flow. The laminar flow is the same. In hydrodynamics, we say that a laminar flow is that kind of layer which all the cells are aligned in a column or in a line. In comparison to turbulent flow, that means that all the cells are randomly distributed. So this is really important because the laminar flow is the only thing that preserves and keeps this single column during the acquisition. If that system doesn't work, we cannot put the cells in a single column to arrive to the interrogation point. That's because it's really important to have a very specific system of pressure working properly during all the process. Like I say, the cells, when they're exposed to the laser, all this luminic information needs to be transferred to filters, lenses and lasers and detectors, basically. So all this optical system, what it does to collect that information, the luminic information, and transfer to electronic information. And all this electronic information goes to the final component, which is the software, which is able to integrate all the information that we're going to collect from the cells. So here on the right position here, we can see a generic optical system. So in this case, the cells are, they're coming from the upper position instead of the opposite. But at the end, they arrive to the interrogation point. Like we said before, it's in an orthogonal position. The cells are exposed to the lasers, and all this information goes to the optical system, mirror, filters, and detectors. And finally, the luminic information becomes into electronic information in the software. So here we have a representation of a spectral cytometer, which is one of the instruments that we're using at work. This is one of them. The fluidic system we cannot see, but it's a lot of containers that contains literally different kind of fluids to keep that pressure. And then we have the software that allows us to integrate and analyze the data in real time. So how works the cytometer? It's very simple. Basically, the first thing we need to have is a heterogeneous biological sample. This sample can come from many sources. It can be from blood, tissues, or it can come from different fields like ecology, can be water, can be many different kinds of samples. In this case, we're going to select or going to use a blood, which is one of the most common samples. And as you already know, all these samples usually have, or they're composed by many cells. Immunological cells, they have different properties and characteristics. So here, we explain that all these samples are composed by different kinds of cells. We represent this difference with different colors. Let's make, and based on this, we can make like a small example to understand how this works. Imagine that we would like to analyze the blue and the yellow cells. Obviously, these two cells doesn't exist, but we try to put this as an example. So here, the blue cell, which is represented on the bottom left, we can see that on the surface of this cell, we can see different kind of proteins. Every protein, which is different, has a different color. So because all these proteins are expressed on the surface of the cell, we call these proteins extracellular proteins, okay? In contrast, if we have the proteins inside of the cell, in the yellow one, the proteins are located in the cytoplasm, but they can be also located in the nucleus. The key of this is, imagine if we would like to analyze antigens or proteins which are expressed on the surface of the blue cells, but also we would like to analyze also proteins which are located inside the cell. So to do that, we need certain tools which are critical or really important to identify these proteins. For this, we usually use antibodies, which are two different kinds of types. We have the monoclonal antibodies and the polyclonal antibodies and the monoclonal antibodies. The main difference between both of them is that these antibodies are able to recognize specific molecules, called antigens, with a very specific manner. The thing is that the monoclonal antibodies, they usually bind to the antigen in a very specific site, one antigenic site, it's called epitope or antigenic epitope. In comparison, the polyclonal antibodies are able to bind to different positions or different locations of the antigen, creating like a multiple connection, binding from different antigenic sites, okay? So we have this tool. Basically, we call them antigen, but most of the proteins, most of the antigens, are always proteins. Not all of them, but the thing is that antigens are, they're molecules which are appears in the body from infection or something, or exogenous proteins that create a specific response, which are detected by the monoclonal antibodies. But we can use these tools to identify these specific proteins. But the thing is that this kind of identification requires a color or requires a specific way to be detected by the cytometer, because in this way, we don't have any weight, we don't have any specific colors or specific ways to detect this binding by the cytometer. To do that, we need colors to be labeled to these antibodies. These colors are called fluorochromes. A fluorochrome is basically a molecule that when it's excited, the electrons which are inside of the molecule are going to be excited and go to a different or higher level of energy. And when these electrons come back to the basal state of energy, they emit emission of energy, which is detected with a specific wavelength. So basically what happens is any fluorochrome, when it's excited with a laser, for example, becomes excited and they start to emit a specific emission wavelength. So we have two different kinds of wavelength. One of them is excitation that allows to excite the fluorochrome. And when this is excited, this fluorochrome is able to emit a different wavelength that can be potentially recognized by the detectors. The good thing is that all these fluorochromes, all these molecules, can be linked or labeled to the antibodies. So here we have a representation of an antibody combined with a fluorochrome. And with these tools, we can identify any antigen of interest, which are recognized for this antibody. And this color, this fluorochrome, is able to be detected by the detectors and filters that we have in the cytometer. So we have that sample, and then we use antibodies, we can identify in a specific way these antigens, which can be located on the surface of the cell or inside. So basically, when we do extracellular analysis, this is when we analyze the proteins or antigens which are located in the surface of the cell, we usually use an antibody conjugated with a fluorochrome. And when these cells are exposed to the laser, the fluorochrome becomes excited, and this signal is detected by the detectors of the cytometer. In addition, we can use a different way, it's called an indirect way, that we can use a primary antibody, which doesn't have any fluorochrome, but this primary antibody can be also linked or binded by the secondary who has another fluorochrome. One way, direct or indirect, is allows to us to identify these antigens in a very, very easy way when the cells are exposed to the laser. With a similar approach, we can use intracellular analysis. This is intracellular, not extracellular. So basically what we do, all the cells, they have a membrane, but when we would like to analyze the antigens which are located inside, we need to permeabilize these cells. So we create small pores or small pores. So basically the antibodies can go inside of the cell and they can recognize the antigens which are located inside, okay? But the good thing is that one approach or another one can be also compatible. So we can analyze at the same time extracellular antigens and also intracellular at the same time. So this is a very, really important tool because they can provide a lot of information with a very small amount of cells. So what is the function of a cytometer? The cytometer has a specific function is to analyze different cell populations from heterogeneous sample, basically. So we can go from the most general to the most specific properties and characteristics of the cells. So we can also stratify or classify the cells according to the level of expression of different markers. Basically, every cytometer has the property to analyze what is called morphometry. So we can analyze size and complexity. Size, that means cells can be bigger or smaller, and the complexity is according to the granularity of the cells. These two properties are based on diffraction of light. So what happens is when the cells are exposed to the laser, they emit a shadow. That shadow is detected for what we call it light scatter parameters, which are mostly focused in identification of the size and the complexity. And according to that shadow, if the shadow is bigger or smaller, they can determine the size of the cell. If the complexity of that shadow is bigger or smaller, they can also detect that. So that's why, because we can identify different types of cells only based on the morphometry or morphology of the cells. In addition, cells can be also analyzed according to the fluorescent properties, including autofluorescent. So some cells, they usually have a spectral signature as a consequence of the presence of the certain enzymatic or molecular chemical compounds inside that emit a fluorescence as well. So the fluorescence can be inherent to the cells, and when this happens, we call autofluorescence. Also can be induced by the antibodies conjugated by the fluorochromes. So because we put antibodies conjugated to be exposed to the laser. So the antibodies recognize the proteins on the surface or inside, then are exposed to the laser, and then finally this information goes to the computer and to the to the informatic of the computer system to interpret the data. There's a different kind of subunit, slightly more complex than the cytometers, which are called cell sorters, which are able to do exactly the same like a cytometer, but the main difference is that these instruments are able to isolate specific cell populations. So we can do exactly the same identification of the cells based on morphometry and fluorescent properties, but also we can isolate cells. So imagine that we have the same heterogeneous sample with the different cells with different colors, and we want only to isolate the blue cells, okay? So based on the antibodies that we used before for the extracellular markers for these specific cells, then we can say to the machine or the instrument to sort specifically the cells. This is really important because we can study and analyze these cells in a very independent way from the heterogeneous samples and can be used for many, many, many analyses. We can do transcriptomic analysis, we can do cell culture, we can do stimulation, we can do many, many things from this population, okay? So here we can have a representation of a spectral cytometer. In the right side, we have a PD fax area, which is a different, it's a cell sorter, it's a different instrument, okay? So the flow cytometry is many multidisciplinary. It's a discipline that have applications in many, many fields. And this is really, really important because they have a direct specific application, mostly in biomedicine, where immunology and hematology are the most predominant areas where these technologies apply every day. In hospitals, to develop diagnostic kits or follow-up treatments for many, many, many reasons, and also for clinical research as well, clinical research. So then we have different applications in industry, mostly in food industry, biotech, or even pharma, with different purposes, and it's happening also in daily basis. Many, many companies, they usually use flow cytometry because it's a cheap and very quick method to analyze different kind of products, raw products or final products before to be ready to the market. In addition, flow cytometry have many, many applications in ecology as well to determine water quality, processing of water or biodiversity. And something that's already quite trendy is analysis of immune response in conditions of low gravity in the case of space. But this is only the beginning, where many, many applications as well to be developed in the space environment. So the flow cytometry market size at the moment include academic research, hospital, clinical testing laboratories, big pharma, and biotechnology companies, most of them. Many of these companies are also based in Oxford, but others are located around the world. Actually, the flow cytometry market is estimated in 2023 in $4.7 billion, and is still expanding. So the applications in medicine are quite relevant, especially in immunology and hematology. And the main reason of this is because all these technologies are applied for many reasons: diagnosis, clinical research. So here we have a very, very basic panel. A panel is composed by different antibodies, okay? And they have the purpose to identify specific cells with a very specific purpose. For example, here we can see that all the antibodies that we have, lambda, kappa, CD8, CD4, are addressed to very specific target cell populations. B means B cells, TC means T cell cytotoxic, that express also CD8, NK means natural killer cells with the specifics 8 marker, TH means T cell helper, mono means monocytes, L means lymphocytes, monocytes or neutrophils, T means T cells, NK natural killer cells, immune cells is CD45, and B cells CD19. But the important thing here is not to memorize these things. The key is all these antibodies are specifically addressed to very specific antigens and proteins on the surface of the cells. And thanks to all of them in combination, we can classify and stratify different populations. So here you can see a representation of the antibodies with different fluorochromes, okay, different colors. The names of the fluorochromes are here. We have many of them in the market. That put use the most common. And the key of this is that we can use a heterogeneous sample like a blood sample, but imagine that we're working with pediatric samples, okay? From pediatric samples, we cannot get too much volume, okay? Because we cannot get the same amount of volume of samples from an adult, a minor, right? So in this case, we usually use a very low volume of sample, around 100 microliters. This means microliters, which is equivalent to 1 or 2 drops of blood. It's almost nothing, almost nothing. So we collect the sample from someone, or maybe a pediatric sample. Then we prepare all the cocktail together, okay? And we put all the antibodies together in the sample. So after the incubation, we can then acquire the sample in the cytometer and analyze the data. All this process to identify all these cells and characterize all of them independently is called immunophenotype. Immunophenotype is one of the most relevant tools in clinical research. It's one of the most relevant methods used today to identify and characterize any target cells in a qualitative and quantitative way. In a qualitative means that if we have the presence or absence of the protein, we can detect that based on the expression of these proteins, and we can quantify how much is expressed. Within a heterogeneous sample, it can be from the most generic to the most specific. And like I say, it's very, very useful in screening studies, in clinical research, in diagnosis, follow-up, and it's really, really relevant. The key is based on this information, we can determine if someone has a specific illness, or if someone has a specific condition, clinical condition, because we can identify all the populations, and if these populations has any variation that could be considered aberrant or pathological. So the way that this thing works, basically, is very simple. Imagine that we acquire a sample of blood. So we have, if you remember, we have 2 parameters to detect the size and the complexity. The light scatter parameters, forward and side scatter. So in the forward, everything that goes to the right increases size, okay? So everything that goes to the right is bigger, everything that goes to the left is smaller. Same thing happens with the side scatter. Everything that goes up is more complex, everything that goes down is less complex. So here in this bivariant plot or dot plot, because we compare 2 parameters, we can see 3 different populations. We can see, for example, lymphocytes, monocytes, and neutrophils. As you can see, the neutrophils are more bigger. That's because they are more to the right, but also more complex because in a higher position in comparison to monocytes or lymphocytes, okay? The cells which are located here that we don't include is called debris. So normally the cells which are very, very, very small and the complexity is very, very small are fragments of the cells. So we are not interested to analyze this. So the reason because we analyze this data, this is a whole blood sample. Imagine that we take 2 or 3 drops of blood from someone, and then we analyze all the surface markers that we saw in the blue cell before. So here, for example, we have many markers. We have D45, it's an immunological marker. So all the cells that express this marker are immunological cells. For example, here, everything that goes to the right is positive for this marker. So we have 3 different levels of expression. The red cells, which are the lymphocytes, has a higher expression of CD45 because it's going to the right. The monocytes, which are in green, which are here, the expression is slightly lower because are going more to the left of this marker. And then the neutrophils has lower expression of CD45 in comparison to these two. But all of them are positive because our negative is here, see? So in this case, we can identify cells which are immunological. Same thing happened with different markers from different populations. We don't need to be in detail, but essentially I would like to explain you how this thing works more or less. So basically, we have different markers. For example, CD19 is a marker of B cells. You can see most of the markers has a CD, which means cluster of differentiation. That means when they designed it and when we have characterized antibodies against these populations, it was defined in a very specific workshop, and they call it CD1, CD2, and every CD represents a specific antigen recognition. So for example, here we have another one, CD14, which is a marker of monocytes. So it's the same population. Same thing happened with different neutrophils, different populations of monocytes, T cells as well. Basically, at the end, what I would like to tell you is that after few drops of blood, we can analyze many immunological cells at the same time, in real time. So imagine 2 drops of blood, and we can analyze many, many, many markers at the same time. But why the immunophenotyping is so important? The reason of this is because using this approach, we can monitor many populations, from healthy individuals with different ages or different conditions, and we can define the baseline or reference values for these specific populations or specific individuals. So if we know what is normal, which is healthy, then we can identify what is aberrant, pathological, or even clinical. So, for example, imagine that someone doesn't have the expression of CD19, we can say that a person, because these cells are lymphocytes, we can say, okay, the cells are, maybe you heard the word lymphopenia. Lymphopenia means that you have lower number of lymphocytes. But if you have lower number of B cells, we can say B cell selective lymphopenia, for example. So we can identify based on the percentage of the cells if someone has expected values or not for a specific population. That's because, for example, other case will be if someone has a lymphocytosis, that means that the number of lymphocytes are higher in comparison to the expected or reference values. We can think, okay, maybe that person has an infection or which populations are more predominant. So that helps us to understand the context of a disease. They can help us to understand any treatment and the follow-up. And also they can help us, and this is really important, to identify new markers associated to a specific disease, which is called biomarkers. Specific diseases cannot be correlated with a specific absence or present, highly present of certain populations. And this can help us to define and create new potential diagnostic tools that can help us, many, many people. So this is one of the most important applications in medicine, immunophenotyping. But like I say, flow cytometry can be applied to many, many, many applications. For example, in industry, one of the most important products we usually can find in the supermarket is a milk. Okay, so the food industry has a heavy weight in the economy. So most of these products require to be submitted or under very specific controls before to be sold in any supermarket. So people, what they usually do in the companies is to take a volume of the milk. This milk can come from the raw product or processed final milk products, and they can try to identify which is the amount of bacteria that they have, okay? Because they have a specific threshold limits that if for some reason this limit is higher, that product cannot be sold in the supermarket. So what we do is to take the same volume for flow cytometry to identify this bacteria and to determine if the bacteria is under this level, or we don't, obviously we don't need to have any bacteria, or at last, no pathogenic bacteria. But the thing is that we usually compare two methods. One of them is a flow cytometry, and another one is microbiology. So what they use is the same product of milk, raw milk or processed final milk, in different plates, and they count the bacteria and then correlate the data that they have in the microbiology compared to the flow cytometry to validate the results. This is really important because they can help us to identify the presence of pathogenic bacteria in final products or in primary raw products in the processing of them. And also they will help us to define and create new sterilization methods to ensure that the pathogenic bacteria are removed before to the final product is released. So there are different methods, heat inactivation or any others. So these kind of methods, which are very, very quick and very, very fast, can help us to determine which is a quality control for this product in any part of the process of the food chain, okay? Then other applications that we can see in other companies is biotechnology. With the biotechnology, we can see most of the companies are focused in the drug development. So here, for example, they try to develop a drug against Plasmodium falciparum. Plasmodium falciparum is a parasite, okay, which is responsible or is it's the agent responsible of the malaria. So here, what they did is to analyze, you remember the size and the complexity of these parasites in the flow cytometry, so we can identify, you can see it's very complex, okay? But the thing is that this dot plot, this representation is also magnified to see much better this parasite inside. From these parasites, what they do to identify the parasites in the cytometer, because as you can see, not all the pathogens can be recognized by antibodies because we don't have characterized yet all the antigens against them. So in this case, they have a Plasmodium falciparum, what's coming from a cell culture, and they put a marker which is called cybergreen. Cybergreen is a marker that can be used to intercalate and stain the parasite inside, particularly in his active nucleic inside. So the cells become green, basically, okay? So you can see this dot plot on the right, we compare the cybergreen. So everything that goes to the right becomes positive. That means the parasites are present and they express this protein, okay? This protein is, the parasites are stained with this thing, and based on this expression, if it's higher, you can see most of the right. If it's lower, go to the left. But I would like to see how these cells, how these parasites are able to infect red blood cells. So we put a co-culture of the parasites with the red blood cells, and the red blood cells are identified by a specific marker, which is CD235. If you remember, cluster differentiation 235 is the name of a marker, basically. So here we compare the expression of the erythrocytes and the parasite. So in this red square, we see what is double positive for both, because in this representation, called a dot plot, we have 4 quadrants. Here is for the cells which are negative for both markers. Here, the cells which are only positive for CD235. So that will be the red blood cells without the parasite. Here we have only the parasite, which is the cybergreen positive, but not inside the erythrocytes. And this one in the red one is the parasites which are inside of the erythrocytes. Why is so important? Because we can use this basic approach to measure how many parasites we have in that sample. But also, we can also use this technology to use different drugs. So what we can see, if you're using different drugs, the percentage of the cells with the parasites here will be lower. So you can establish how efficient is a drug based on the reduction of the positive, double positive cells, which are located here, okay? That's also very good because we can define different new ways to eliminate or neutralize the action of the parasite inside the erythrocytes using different drugs, and we can compare this with a very, very basic approach. Like I say, the application industry is quite routinely based and has a lot of applications. This is only a minor, it's only a few of them. Like I said before, the flow cytometry can be also used in different fields like ecology or environment. So this is important because if you would like to analyze the quality of the water from a specific lake or areas where the water can be stank, or the things that we can analyze the ratio of the proportion of the phytoplankton versus the cyanobacteria. So how we can do this by flow cytometry? So as we said before, the forward and size are able to discriminate size and complexity for this kind of particular cells. But the thing is that most of the phytoplankton and also cyanobacteria are not necessarily spheric cells. As you can see in the bottom, most of them are filamentous or different shapes. So based on these two parameters, we also use some parameters related with certain lasers. For example, here we have one of the lasers, which is the blue laser, with a specific nanometer excitation wavelength. And here we are using a different laser. It's called yellow. Well, it's a yellow-green laser, with the emission wavelength is completely different. So when we compare and we make a dot plot of both, we can discriminate the different kind of cyanobacteria and different phytoplankton. The reason is because most of these organisms, they usually have inside fluorochromes. In fact, from most of these, all these cells and algae are the key or the base, the key to develop new fluorochromes. In fact, many fluorochromes that we are using in antibodies originally comes from algae or from this kind of organism. So when we talk about the fluorescence that some cells can have as an autofluorescence, most of these organisms, like different cyanobacteria or different phytoplankton, they have inside different molecules that when they're excited, they emit a signal. So if we can use the forward and size to identify different cells, and also we can identify the different level of expression of the emission of these fluorochromes, which are located inside of the cells, we can stratify all of them easily. And this is really important because if we have the sorting machine, sorted cell, we can also isolate independently all of them. One of the reasons it's really important because the phytoplankton and the cyanobacteria are always really important, because based on the proportion of one of them or another one can tell us, or they can give us information about the quality of water, or basically, and also the biodiversity of this sea or the lake or lakes where the samples are collected. In addition, like I said before, the identification of algae from the sea or from other locations can help us to develop new fluorochromes because they have these pigments inside naturally. Well, another application that we have, and it's also quite relevant, is applications that we have in space. The thing is that the conditions that when someone is in a space flight, especially when this space flight is prolonged in time, the immune response is lower because of low gravity. The reason of this is because the low gravity has an impact in the way that the cells can respond to specific antigens. This is quite relevant because imagine that someone in the International Space Station, or maybe in future colonies at some point, they want to be settled for a long time, and if that person develop any disease eventually, we need to understand if that person can, have a good enough immunological response against specific antigens. So most of the studies are developed in the past. They usually collect samples from the crew of the space flights, and they take samples from simulated gravity, microgravity from a specific time, and also from samples from the same individual, but taken in, with the gravity that we have in the Earth, okay? And then we compare the immunological response against different antigens at different time points, okay? So the reason is that we can try to compare the immunological response in conditions of low gravity and also in conditions of non-gravity. The thing is that we try to understand what is the mechanism behind, or what's the reason, because sometimes the cells in low gravity doesn't respond properly to specific antigens. So because imagine that someone has any flu or any infection, anything. So we need to try to understand how we can neutralize and try to attenuate or mitigate their symptoms and improve the immunological response in these conditions, because that people cannot come back to the Earth easily. So here in this, in the International Space Station, in the US lab module, they develop a very specific cytometer to analyze the immunological response. The main problem is that the cytometers that we know, they usually work in normal gravity conditions, right? So the things that to create that kind of laminar flow inside to align the cells, we need to recreate a different mechanism to analyze the samples. So they develop a new cytometer called Microflow 1, which was tested in many parabolic flight campaigns. So basically, they take the cytometer in a flight, and they try to simulate the low gravity, so with a high fall. Basically, what they try to simulate or emulate this kind of low gravity during the time of acquisition. They recreate a machine which is able to analyze the cells independently of the gravity, with a very innovative fiber optic able to interrogate the cells independently of this gravity, which is really, really useful. Here you can see one of the astronauts, Chris Hayfield, with this instrument. And like I said, this is really important in case that we have any potential disease, or we're trying to understand how the immunological response can work. And this is really relevant, especially for future colonizations in different locations eventually. So basically, this technology is used for monitoring the immune response of the crew, basically, in low gravity, to understand how it works in these conditions and explore new pathogens. Because imagine that all pathogens that we can potentially encounter in the future, most of them are proteins, and most of them proteins, they usually can have, some of them, antigenic properties. We don't know if in the future we're going to have exoproteins that potentially can create or develop a disease. So it's good to start to understand how potential antigens or new potential antigens, based on what we know, can have an impact in these conditions. Okay, that's because applications in space is something that starts now. I'm pretty sure for most of you in the future will be something more common than from us, but it's something that is start to grow very, very, very fast. So basically, finally, I would like to make a very quick summary about the flow cytometry and how it works. Basically, we can say that flow cytometry establishes a very extraordinary balance in terms of resolution, isolation, accuracy, and speed of any kind of cell population. It's a multidisciplinary method that can be applied to many fields, as you can see before. We can analyze many, many parameters at the same time from a very heterogeneous sample, from the most generic to the most specific, and it's really, really helpful in the study of pediatric diseases because we can use a very small amount of blood or any other sample to analyze and understand how the immunological response works. Finally, like I say, flow cytometry is conventionally used in many, many fields. For food industry, I put you one example with the milk, but there are many, many other ones. And also can be used in veterinary for novel drugs, most of them. In pharma industry is also for to monitorize new drugs for different kind of diseases. In ecology, like I said, can be used for biodiversity and quality of the waters. And finally, in the space, it's really relevant because we can try to understand how the immune response can work and can be also prepared for future and new antigens eventually at some point. So thank you, everyone. If you have any questions, I will be very happy to reply. I hope you understand, and I hope that this seminar can inspire you to work in science eventually at some point. Thank you. Oh, yes. I will check the question. Basically, about the complexity. Someone asked me about, could you explain what you mean by complexity much more? Basically, complexity is a term that we usually use for granularity. So some cells are more granular, more irregular in the surface. So that can be also tell us about some difference about certain cells between other ones. So one cell can be smaller, but also less complex, more spheric, and other ones can be more complex, I mean, with more irregular shapes that can be also considered like a more complex, basically. The thing is that this can be applied for different kind of things. It's not the same like a bacteria, than a monocyte, than a lymphocyte, and can be very easy stratify based on these two parameters. The thing is that it's like I would say about the algae. No? Most of the algae are discoidal forms, not the spheric. So even the complexity is different. There are different ways to interpret this, but basically it's about how the cells can be more granular in the surface, basically. There is another question about how will biomedicine differ if flow cytometry had not existed and how important is in academic research. But basically, most of the methods that we used before, most of them are monoparametric. That means that we only analyze one dimension, one parameter. The flow cytometry is one of the few methods that can be used as a multi-parametric. That means that you can analyze many dimensions, many parameters at the same time. It's something that never happened before. I give you a very small example, but today we're running panels at the moment with more than 30 or 40 markers with a very, very small amount of blood or any other sample. So they can give us a lot of information that any other technique cannot do it because, like I say, if you have maybe something called ELISA or different kind of methods, most of them only can analyze one parameter. Flow cytometry is by far one of the most complex because they can analyze many of them at the same time. The next question is how the pores are created in the cell membrane in order to analyze intracellular proteins. That's a good question. So basically, to do this, there are specific detergents that we can use to generate artificial pores inside of the cells. So there are specific kits that we usually use to put a very specific amount of a detergent that doesn't kill the cell, but they create enough pores to allow the access of the antibodies inside, okay? So depending on the detergent, these pores can be generated on the surface of the cells or can be also inside of the nucleus as well, because the membrane of the nucleus is also inside. Depending of how much we use, we can use this. The only thing is that when you use this kind of detergents, you need to fix the cells, and because otherwise the cells, when they generate the pores, eventually they will close again. So we usually use a fixed cell to fix that staining inside the cell as well, so we can quantify how much is expression inside. This is really important to analyze for example, something called cytokines, which are peptides of low molecular weight that can regulate immune responses. Okay, so these cytokines are really important to understand immunological response because they can regulate how the immunological function can go to another way or another one. Anyone has any other question? I hope, yes, please. Okay. How much research is there about the application of cytometry in space? Okay, we're starting now at the moment. So most of the studies are in two different sites. One of them are mostly in the ground here. So what they do is to bring samples from the space to Earth. So we usually transfer that samples in very high restricted conditions, is very high biosafety controls, because we don't know if that samples that comes can bring any potential pathogen. We don't know. So these samples are inside of a lab. It's called CL3, containment level 3. And that samples can be processed for many, many processes, not only for microbiology or exobiology, which is an application that we usually use. Other samples can be processed in the same station, International Space Station. So they can use this cytometer, the microflow one, and there are many publications that you can find online about the data obtained from these samples. What we see is that at the moment, when someone is in a prolonged space flight, you can see not only the cells, also the bones, the muscles. They start to be not degenerated, but they are more energetic. They have a lower response. So this is really important to understand how the immunological response. That's because most of the crew usually do a lot of exercise and they are monitored all the time, because we know that the prolonged flights can have an impact on this on the immunological response. So they're trying to develop new systems. They can simulate gravity to attenuate that effect. But it's something we're still working on these things. I know many people who are working in the space as well, working that kind of things. And we usually provide also help because we collect samples, not only for the blood, from different fluids, nasal cells, nasal washes, to understand that kind of response, especially respiratory disease. But imagine that someone has, for example, a digestive or something more, conflictive in the space. We need to try to find a way to neutralize the reactions and also to understand how to stop and to improve the immunological response if something happens, not now, even in the future. And the other way to analyze also the other pathway of analysis, trying to identify potential new antigens or exoantigens, because in low gravity conditions, for example, bacteria become more resistant. So we need to be careful about how this can impact in the way that the cells are able to respond to bacteria which are more resistant, or even potential new antigens that we don't know, that we think that most of them presumably will be proteins, because it's the most common antigen. And depending of the level of antigenicity, we can work the way to understand the immunological response against something new that we are not ready yet, but eventually we will. So I hope, any other questions, please? Happy to, if you want any publications about this, I can send to Jack or Pravahi more publications about this, if someone is interested. Okay, I hope everyone like. Another question: how frequently is flow cytometry used to analyze patient samples to improve medical care? Is just to confirm suspicious of a specific disease, or use it to count cells in a standard blood test? Okay, so it's very common. Basically, in all the hospitals, we usually have two different teams. One of them is for clinical research, and other one is for routine. So basically, for routine, what they usually run is panels all the time to determine the percentage of different populations all the time. So we have reference values for different cohorts of population based on age or different conditions. And when someone has, for example, higher or lower percentage of certain cells, that limit is highlighted to the clinician, and we say, okay, that person has lower population or higher population that could be compatible with a specific condition. And in particular for medical care, it's really relevant because especially people who are under specific treatment, we can use in the same panel to see how that person can improve versus time. So we can see if specific populations are responding properly to a very specific antigen during different sessions of a specific treatment. And also can be also used for to confirm a specific disease, because like I said before, you have there are specific biomarkers associated with a specific disease, and when that happens, when you have a very low expression of something or a low number of cells for a specific population, we can say this diagnosis is compatible with this potential disease. So then the clinician will run additional tests to confirm with different other methods if what we see is true or not. So the thing is that to define what is normal on the standard values are highly, highly important, because they will give us the tool to understand how the expected values are, and if something is aberrant or they have any clinical consideration, we can detect very, very easy. And one thing that we're doing at the moment, we all, I mean, in general, all the networks, we're creating a very huge databases online from many, many, many people, patients and also healthy volunteers. And when a sample arrives, we can identify much better if the sample is compatible with a specific disease or not. So I think, Jack, you want to say something?
Just to say that was excellent, really. We have run out of time, so we have to end things there. But it looks like we got pretty much everyone's questions on here, which is really good. So yeah, thank you, Jesus. It's been fascinating listening here in the background, and clearly the audience have really enjoyed it from the quality and the number of the questions. So yeah, so thank you, everybody. Remember that they will be going online, so you'll get an email anyway, but just in case you don't see it or whatever, they will all be online. So if you, like I said, there's one that you've missed or you want to catch back up on this one and make any notes, you'll be able to find them on our YouTube channel. But yeah,